Mindsetters, welcome to today's Grade 12 Life Sciences show. Today we have a special revision lesson in which we'll be doing exam questions for paper 1. We have selected highlights from lessons shown earlier in the term to help you revise this term's work. So please post your comments and your questions on our Facebook page or on Twitter at LearnExtra so we can help you with your revision. You can also download the show notes for today's lesson from learn.mindset.co.za or click on the link on our Facebook page. Now it's time to get on with today's lesson.
The first one adapted from Gauteng, June 2014, question 3.3. As you can see from the diagram, or before we even go there, the diagram below shows a cross-section through the ovary of a human reproductive system. So it already tells you that we're dealing with reproduction. And guys, this is something you need to also get into your head, that when you look at a question, the stem will already tell you either which chapter it is, or the diagram will be a clue. So you need to put yourself into context. Okay, right, this is about reproduction. This is my body of knowledge in reproduction. Let me use that knowledge to answer the question. So let's look at that now. The question says, answer the questions based on the diagram. They're giving you a diagram there of the ovary and 3 labels A, B, and C. And the first question asks you to label A, B, and C. And obviously, the first one, A, is the Graafian follicle. The Graafian follicle. B would be the ovum. And C is the remainder of this Graafian follicle after the egg has been released, and that would be the corpus luteum. By the way, the question only asked for A and C. Aslam already gave one extra answer, but that was good because it's going to come back later to bite us anyway. But remember, follow instructions. This question asks you for A and C, not A, B, and C, not A to C, but A and C. So you would have answered it accordingly, only A and C. Identify structure label B. We were forward, we were precocious, and we have answered that already, and that is obviously the egg or ovum. Right, the right terminology, ovum. Give the ploidy. Ploidy. Now, in your studies, where did you hear about ploidy? This word ploidy, where does it come from in your studies in grade 12? There's only 2 words that you have learned with this ploid in it, and that would be haploid or diploid. So, is this structure B haploid or diploid? This is what the question is asking. And obviously, because it's a gamete, it would be haploid or N. Give the name of the hormone secreted by structure A. Structure A was the Graafian follicle. So the hormone that is only one hormone. Notice it said the hormone. It's not hormones, one hormone, and that would be estrogen. Hormone, estrogen. Provide one function of the hormone identified in question 1.3a. What is the function of estrogen? Firstly, in the females, the first function would be to bring about secondary sexual characteristics in the female. Okay, secondary female characteristics, and more importantly, well, I can't say more importantly because they're both important, but in terms of our study of reproduction, this is core. It stimulates the thickening of the endometrium. In other words, it causes the uterine lining to become thicker. Why? For possible pregnancy, for possible implantation. And sometimes it also it also stimulates the production of LH. When estrogen is high, LH will be secreted after that. Discuss how oogenesis is prevented once a female is already pregnant. Oogenesis prevented when a woman is pregnant. Which hormone is high in concentration when a woman is pregnant? FSH, estrogen, progesterone, or LH? You got it? progesterone is the one that's high. And what do we know about progesterone? A high level of progesterone inhibits FSH. In other words, when the levels of progesterone are high, FSH will not be secreted. Now, let's think for a moment. What is FSH? Follicle stimulating hormone. So, if the follicle stimulating hormone is inhibited, it will not stimulate the follicle. Good, you got that. And if it doesn't stimulate the follicle, then oogenesis cannot take place. Because where does oogenesis take place? In the follicle. Good. So let's look at that answer again. When she's pregnant, the progesterone levels will remain high. Key is not about that there is progesterone, but the progesterone is high. You must mention that. Those two must be tied together. This high progesterone inhibits the secretion of FSH. And it will not stimulate the formation of ova or the follicles, and therefore no oogenesis will take place. So in other words, this was another way of asking you questions on reproductive without just telling you to write an essay on it. My finger seems to be working better than a pen today. Okay. Now, also from Gauteng, records of human fertility for the period 1941 to 1990 have shown changes. We need to worry about this one. Have shown changes in the sperm counts of normal men. The table below summarizes the changing percentages of men with high or low sperm counts over a period of 50 years. The changing percentages. So there's a table, 1941 to 50 and so on. And they're telling you here in the first column here, men with high sperm counts, the percentage was 50, then 45, 28, 21, 15. You can see what's happening there already. And on this side, men with low sperm counts, 4, 4, 5, 11, 14, 18 in 1990. And we are now in 2014, so we need to start worrying. On the same system of axes, draw two sets of bar graphs to compare the percentages, not the sperm count, but the percentages of men with high sperm counts with those with a low sperm count over the 50-year period, 1941 to 1990. And we're not going to waste too much time. Just going to open that up. So the heading, the title, must have both variables. And the variables are, one, the percentage of men with high and low sperm counts, and the period from 1941 to 1990. So over time. Okay. The x-axis heading, time period in years. Notice the unit must be given. And here, because you're using the word percentage here, then that's the unit on its own already. Percentage of men. Okay. If you want to, you can put that in brackets there, percentage, but it's given there already. Notice there's a key also. The one that is shaded, high sperm count. The one that is unshaded, low sperm count. So there's a mark for that as well. Okay. Notice the intervals, 10, 20, 30, 40, 50. They are equidistant apart. And the bars, the width of the bars are the same. And the distance from one set of bars to the other set of bars is also the same. These are all graphing skills that you need to have. Obviously, the plotting is easy. You need to just plot them according to what the table gave you, and you will then get your marks for that accordingly. There were 7 marks given there. All right? The 1 mark would have been given there, 1 for this whole idea here and the interval, 1 for this whole idea here and the interval or the bar's width. That is 1, 2, 3, 4, and you would have got 3 marks sometimes for, oh no, 1 mark for the type of graph, and 2 marks for plotting sometimes. And that will give you 7 marks. Good? Describe the trend for men with low sperm counts and compare it to those with high sperm counts. The trend that you saw, and I highlighted that to you on the table. You should have picked that up already. Without the graph, you would have picked it up with the table already. As time progresses, we're comparing it with time. There's 2 variables. One is time and one is the percentage of men. So that's what you have to do. And generally when you're describing a trend, you must start with the independent variable. You can't say as the percentage of men increase, so too does the time. Doesn't make sense because the number of men is not affecting time. Time is affecting men. Okay? So as the time progresses, the percentage of men with low sperm counts increases. It doesn't say that the sperm count decreases. That's not what it's saying. It's saying that the percentage of men that have low sperm counts, it increases. And the percentage of men with high sperm counts decreases. Lots of learners, and this one here would have been a ridiculous one to start off with the number of men. But in other examples, lots of learners, they switch the data around. So they say, for example, let's say enzyme activity and temperature. We know that when an increase in temperature occurs, the enzyme will become denatured, so therefore its activity will decrease. Now what learners do, they say as enzyme activity decreases, the temperature increases. Now that can't be right because the dependent variable is dependent on the independent variable and not vice versa. So in other words, the enzyme activity is the dependent variable. It depends on temperature. Temperature does not depend on the enzyme activity. Hence, it's called the independent variable. So that's where we have this answer here. Lots of learners confuse it just because of the independent and dependent. Remember, whenever they ask you the relationship between two variables, start with the independent variable because it is that variable which affects the other variable.
I think it's time for a break now, so don't go away. We'll be right back. Welcome back from the break. Let's not waste any more time and get straight back to today's revision lesson.
Study the graph. We're still busy with reproduction. I've kept them in sections so that we do one section and we've covered both sides of the spectrum now. The next one is if they give you a graph, and this one was done in KZN, question 2.3. Study the graphs below showing the levels of the hormones involved in the menstrual cycle in most women. So there we go. We have this graph, famous graph that we normally see, more or less, not always exactly the same, but at the bottom we have the days there, and in this block we have the ovarian hormone levels, and in this block we have the pituitary gland hormone levels. So we're getting two sets of hormones. You need to know which hormones are secreted where, and I'm just going to put here our famous word FOB, and those are the hormones that you need to concentrate on, but you need to know which ones are secreted by the pituitary gland and which by the ovary, in the ovary. Okay? So they're giving you the story there. We're seeing what's happening there. There's the hormone A, B, C, and D. And there's your day 14. There's the beginning of the cycle, middle of the cycle, and end of the cycle. So let's get into the questions. Provide the label for hormone A. Now, to identify hormone A, you have to look at what I told you here. What did we write there? FOB. And if you remember, FSH, estrogen, LH, and progesterone. Why do we write it as FOB and not FLOP? Because that is the order in which these hormones increase in a menstrual cycle. So we look at the one that increases first in the cycle. We look at the one that increases first in the cycle, and that would be this one here, that's high, that's B. And then this one here, A, peaks in the middle of the cycle. You're looking at it where it's increasing. After B increases, you see there's an increase in C, and last, there's an increase in D. So based on where we found the increases, we can place them. That B will be FSH, C will be estrogen, L will be LH, it's A here, because it peaks in the middle, 14th day, and the one that peaks later, D, would be progesterone. So once you've identified that, that is why this hint and clue is always important for you. So you write that down and you identify it already. You can write it on the graph or whatever. So they're asking for hormone A, and already we know from our identification that that's LH, luteinizing hormone. According to the graph, during which period of time is the level of hormone C lower than that of hormone D? When is hormone C lower than D? If we look at this whole period from 0 to 14, C is higher. You do agree, I'm hoping? All of that period there, C is above D. Till here, it is still above D up to that point. And from here onwards, we find that it is below up to the 28th day. So it's somewhere in between 14th and 21 days there and the 28 days. So that's the period that we are looking at. Again, you didn't need to be a rocket scientist to figure that out. You had to read from the graph. From the 16th or 17th day to the 28th or 29th day. Name and explain the relationship that exists between the hormones labeled B and D in the menstrual cycle. And I'm not going to waste a lot of time discussing this. I'll tell you why just now. B, FSH, and D, progesterone. The relationship. This is the relationship we just spoke about in the last question. Same thing. Notice the question is different, but the answer is the same. First of all, they ask you to explain the relationship. This relationship is called a negative feedback mechanism. You needed to say that first. Thereafter, you say increased levels of progesterone will inhibit the hormone B, and therefore it's going to decrease the number of follicles. Or you can say then when the levels of D progesterone decrease, the B stimulating hormone will be released from the pituitary gland because it's a negative feedback mechanism. So you're explaining the relationship. Okay? Describe the changes that occur in the ovary during the 28-day cycle. Now, without this graph, you could have answered this question because it's just telling you to tell us what happens in the ovary. In the 28-day cycle. But you had a graph to help you. In the 28-day cycles, follicles are released. Follicle grows and matures into the Graafian follicle. On approximately the 14th day, the ovum is released. All right, they're writing it there. Ovulation takes place. The remains of the Graafian follicle becomes the corpus luteum. Description, you're just describing the events that are taking place under the influence of LH. The corpus luteum, you can even say that secretes progesterone. You can add that. But remember, you're not going to talk about the action of progesterone because they did not ask you what's happening in the uterus. Okay? And the corpus luteum secretes progesterone. If fertilization does not occur, it degenerates quickly. That's about it in a 28-day cycle. Especially in this 28-day cycle, what is happening to progesterone in the end? Can you see what's happening? It is decreasing. If progesterone is decreasing by the 28th day, it would mean that this woman is not pregnant. That is why, because corpus luteum has degenerated, progesterone levels have decreased, and you can see what's happening to estrogen there later. It will start increasing as well. And look at FSH there. FSH first. There. FSH starts increasing, and then estrogen will start increasing thereafter. Okay? So you need to be able to see these clues on a graph, and you'll be able to determine whether a woman is pregnant or not. Good. Come on. Hey.
Bit of a love-hate relationship with the watch.
Yeah. Back and forth. Okay, we still with the same question. If fertilization occurs on day 15, so in other words, after the egg is released and fertilization takes place, describe the changes that occurred in the fertilized egg until the embryo attaches to the endometrium. Again, a description of a question. You're only going to describe what's happening to the fertilized egg. First of all, the zygote will divide by mitosis to form a solid ball of cells called the morula, the many-celled stage. The morula, further division takes place, can become the blastocyst and blastula. There's much more to it. There's more intricate details in this process. But for your exam, this is the detail that you need to go. This is the depth at which you need to go. The blastocyst or blastula develops into the embryo, which then attaches onto the endometrium or becomes implanted onto the endometrium. That is what, up to that point, they needed you to explain. A woman takes a temperature reading on a daily basis during her menstrual cycle. The table below shows her temperature records for days 11 to 19 of one of her menstrual cycles. A slight drop in temperature followed by a sharp rise in temperature indicates that ovulation has occurred. A slight drop in temperature followed by a sharp increase, rise in temperature will tell you that. So day 11, 36.5, 36.6, 36.6, 36.6, 36.6, 36.2, 37, 36.9, 36.9. So you need to look at that. According to the graphs, on which day do most women ovulate? According to, not the graphs, but the table in this case. It would be day 14, around there. According to the graph, sorry, we could go back to the graph there. The other one there that said the graph would say that most would be on 14. And according, some women use the information in the table to prevent themselves from falling pregnant, use the information to explain why this method of pregnancy is not reliable. Now look at this table. We said that most women ovulate on day 14. Here, what do we notice? When does the temperature drop? It drops on day 16 and rises on day 17. So this woman's ovulation probably took place there. Okay? So let's see what we have to offer there. The woman ovulated on day 16 or 17. We could have seen that there. Indicating that the days of ovulation can vary from woman to woman. And in the same woman also. 1 month it may be on the 14th, the next month it may be on the 15th, and so on. There's so many other factors that can happen. Stress can cause it to be the ovulation to take place quicker and so on. And if they avoided intercourse on day 14, she could still have fallen pregnant after that because ovulation took place after that. And anyway, just remember, the sperm can last in the woman's body for up to 72 hours. The sperm can remain alive for up to 72 hours. So the chances of playing with that, if you do not want to fall pregnant, not a good choice. Okay, let's move on to see something now. And we go to Gauteng, June 314. Diagrams 1 and 2 show a section through a portion of a human eye. Study the diagrams and answer the questions that follow. Good. There's a clear diagram given to you there. They've labeled two things, A and B. Which diagram represents the eye when a person is reading a book? Now, we have to ask ourselves, which part of the eye is involved in focusing? And reading a book would represent near vision. And your answer would be the lens. What must happen to the lens when we want to read a book? The lens must be more round or more convex. Okay, so let's look at the diagram. It's obvious from the diagram then that the lens on the right, diagram 2, is rounder than the one in diagram 1. There is no A, there is no B in terms of the diagram. There's labels, yes. But the question asks which diagram. And if you give the answer A, your answer is incorrect because there's no diagram A here. There's a label A. Your answer had to be diagram 2. Okay? Name and describe the process that causes the change from diagram 1 to 2. In other words, how do we change the eye to see near? And that's known as accommodation. First we have to name it. Accommodation. Prefer the yellow. Accommodation. So here the ciliary muscles contract, causing the ciliary bodies to move towards each other. They contract towards the lens. And when we were teaching the section, I stressed that point, that the ciliary muscles contract towards the lens. And the tension on the suspensory ligaments decreases, causing the suspensory, I want to add one part there, causing the suspensory ligaments to become slack. That means they're not tight. And if they are slack, there's less tension on the lens, and the lens then becomes more convex. Let's look at a diagram. Here's the suspensory ligaments here. Okay? Your ciliary body muscle is located here where this circle is. So if it contracts towards the lens, it's not pulling the suspensory ligaments. And the ligaments are not tight, they're not pulling on the lens. So the lens takes a round shape. Whereas in this diagram, as you can see clearly, mind the pun on see clearly and the eye in one sentence, there you got your suspensory ligaments that are taut. The word is taut, T-A-U-T. That's because they, look at the size of that circle now, ciliary muscles. They have pulled away from the lens. So they contracted away from the lens. And when they contract away from the lens, they're pulling the suspensory ligaments tight or taut. And when the suspensory ligaments are taut or tight, they're pulling the lens, you know, China eyes, you want to call it. Just now somebody says, I am what? Homophobic. All right, let's move on. Discuss how structure B is suited to its function. What is structure B? It's the outermost layer of the eye, the white of the eye, and this is known as the sclera. When they ask you suitability, you must first know what we're talking about. The sclera's function, we must also know, is to protect the eye and to maintain the shape of the eye. And to do this, what does it have? The sclera is tough and non-elastic. Oopsie. Sclera is tough and non-elastic, and therefore helps to protect and to maintain the shape of the eyeball and provide protection there, right? An unequal curvature of structure A can cause visual problems. Unequal, the key is unequal. Structure A, again, let's go back to structure A, is the cornea, that part, the cornea. So they're saying unequal curvature. And the word unequal is the clue here. That means the person is suffering from, you guessed, astigmatism. That's correct. How can we treat it? We use glasses or contact lenses or laser strategies. And when we're using these glasses or lenses, these glasses or lenses must be unequally ground. They must also be unequal to compensate for those parts that are lacking on the cornea of that person. That's what we have to do there. Good? All right, another question on the eye. Provide labels for A, B, and D. Again, we've discussed this already, so it's going to be easy. Ciliary muscles, A. All right. Suspensory ligaments, B. And D is the cornea. We discussed that just now in the last diagram. Write down the letter and name of the part that provides color to the eye. That would be the iris, and the letter would be C in this diagram here. Iris. Provide the term used to describe the shape of part labeled E. Labeled E is the lens. In this particular diagram, that would be more convex, biconvex. Which diagram 1 or 2 represents the eye of a person looking at an object that is closer? That's easy again. It would be diagram 1 in this case. Notice the questions are similar. Two different examiners. They haven't spoken to each other, but similar questions coming out. This is the need for going over the papers, because this is the way that you have to learn for your work. Is the refractive power of the lens smaller in diagram 1 or 2? Refractive power, is it smaller in diagram 1 or 2? Obviously, the rounder the lens, the more its refractive power. So your answer is diagram 2. A rounder lens has a greater refractive power. And with that, we go for the break.
Okay guys, I hope you're enjoying this revision session. If you are struggling and need help, please post on the page or send your questions to the Learn Extra Help Desk. The links are on our Facebook page. But now it's time to take a very quick break. Don't go away, we'll be right back. Welcome back from the break, guys. Doing revision is really important, so I hope you are enjoying this lesson. I'd love to chat to you on our Facebook page or on Twitter, so please let us know how you are doing. Enough of the chat, now it's time to get back to some more revision.
The first question, guys, says the diagram below shows the various stages in a life cycle of a human. So they're telling you here's a male, here's a female. Stage 1, body cells. Stage 2, gametes. So it'll be the same for both sides. Stage 3, something is happening there to those gametes. And we're having this situation here. And stage 4, it become body cells again from there to more body cells there. State the chromosome number of the cells represented by A, B, and C. Right, remember first of all, what was the clue? This is a
Human.
human. So Tora,
23.
in A,
23.
is right, Yunus?
I disagree.
You disagree. Tell me why.
It's
Because
still
this is a
body cell.
body
not
cell.
yet
This is a
an-
normal, or another word is a
Somatic.
somatic cell. And somatic cells have
46 chromosomes.
diploid, isn't it?
Yes.
Diploid means the full complement. So therefore, Yunus,
It's 46.
46. Good. And what the next one was B. B is a gamete. Now Tola, what is that?
23.
That will be 23. Why? Because this is a gamete.
Gamete.
And gametes have
Half the number.
Haploid.
haploid chromosome number. Why, Eunice? How come it became
they
up?
undergo meiosis, which
Because
is given-
meiosis takes
Yes.
place in between to give rise to that. Excellent, excellent, guys. Or lastly but not least, C. Now that comes together. Now, Tola?
46.
46 again. Excellent, good. Right, let's go there. So A was 46, B was 23, and C was 46. Please, the answer is not the thing here. The reason why we came to this answer is more important. And Tola opened up with an incorrect answer, which was excellent for us all because it gives us a chance to explain why the answer is incorrect and how we go through that. Good? So, by the way, just so that you know, we're talking about meiosis in this question, right? So from what we were talking about earlier.
Yes.
Name the structure at stage 3. Stage 3 is this thing here. What is that?
Can it be an embryo after fertilization? has
Immediately,
occurred?
what do we call it? Immediately. When these two things mix, what do we call it first?
Fertilization.
No, the resultant structure.
It's fetus.
No.
Zygote.
Zygote, excellent, that's it, good. It's a zygote first, and the zygote will then, Eunice, become an embryo
Embryo.
and a fetus eventually, and then it'll be the baby, right? We got it?
Yes, sir.
Between which two consecutive stages does meiosis occur in the life cycle?
Meiosis. And
Remember, we got stage 1, 2, and 3. Consecutive means one after the other. So between which two consecutive, I've already given you the answer anyway. Which one?
1 and 2.
1 and 2, because I wrote it there, right?
Yes, sir.
So it's between 1 and 2. Notice the language is important. You need to listen to the language as well. Between which two consecutive stages does mitosis occur? Now that's easy because we've already answered the first one.
I'd say C and D.
Or they ask for stage, remember.
Stage 3 and 4.
Although your answer is correct, but notice mindset is, when we say C and D, the examiner can say, I didn't ask you for C and D. I asked you for which stage. So be careful, all right? Even though the answer is correct. Maybe they give a concession and they give you for C and D, but rather be safe than sorry. So it will be between stage 3 and 4. Are we covered with that one?
Yes.
I didn't do too much on meiosis because we'll cover more meiosis questions in paper 2 revision as well. All right. This one here now says, the HSRC conducted a survey on food security. So which section of our work?
The human
Human
impact.
Human impact, right. So they're giving you all the stories here. I'm not reading all of that. They're giving you different provinces and they're telling you that in these provinces, there are so many % of houses that are food insecure. What does that mean, food insecure?
They're wasting food.
It is not going to last long.
More importantly, they don't have food, right? Not necessarily that they're wasting it, but they don't have the food. So these people don't have the food. Maybe because people are wasting,
Wasting.
right? What is meant by food security? Any one of you?
Conserving the available food for the future.
Right.
Such as saving.
But if I'm asking you in this room here now, if I say what is food security for people in this room, it would mean that all of us
Have food.
have food constantly, isn't it?
Yes.
All right? So food security would be having access to enough food on a daily basis. That means I can't have lots of food today and tomorrow I got nothing. It must be on a daily basis. Why? So that we can live healthily. Use the data and table to draw a bar graph for the 4 provinces that had the high. Now I can't ask Tola to do it from there or Yunus to do it from there. So I'm just going to open the graph and we're going to rush through this one because over the last few weeks we have dealt with the skill of graph drawing. Remember, the graph must have a title there and it must have both the variables, percentage of food insecure households in 4 provinces. Okay? It must have a heading on the y-axis and the x-axis. Provinces and food insecure households. Because it's a bar graph, the width of the bars must be the same. And the distance between consecutive bars must also be the same. Okay? And then when we're going up here on the y-axis, because we're using a value, the value must be in equal increments. 5, 10, 15, 20, and so on. So that's what you would have drawn as a bar graph. State how the use of fertilizers by farmers can increase food security for a country.
It can increase the
Must
growth.
talk louder, right?
It
I
can,
can't hear you, I'm a bit deaf.
it can increase the rate, the growing rate of the plants.
Good, it will increase the growing rate of the plants, and because of that, that means there'll be more food. Because you're going to give, fertilizers are going to give nutrients for the plants. If they give more nutrients for the plants, there'll be more plants growing, and because of that, there'll be more crops. More crops will mean more food. How does it decrease, Eunice?
Some fertilizers can be harmful.
Yes, so how come?
They can cause the food to be not okay in a way, such
Right.
as genetically modified food, which is not.
Don't confuse the two, right?
Okay.
We're not talking of genetic, we're talking about fertilizers only. Right? So it may compromise the quality of the food somehow. This is what you're saying, generally. Okay?
Yes, in a
So
way.
that's one reason. Any other reasons you can think about, Tola? No? Okay? One is that they are expensive. Fertilizers are expensive. Okay? And it can cause the food price to increase. If the food price increases, that means people can't afford it. So that's no good. All right? Remember, when a farmer is using fertilizer, he has to now add that fertilizer that he's using, the cost of that fertilizer to his product. And so you and I are going to suffer because of that. And what Eunice was saying, overuse of fertilizers can cause oxygen deprivation in the soil, which will eventually reduce crop production. So it will cause harm into the soil, and this in turn will decrease the amount of plants that's going to grow, and if that happens, we'll be food insecure. There'll be less food available. Remember, guys, don't just memorize an answer and chuck it out there. You can get caught with that. You must understand what we're saying. There's, again, steps that we are talking about. So the first step is oxygen gets less in the soil. What happens because of that? There'll be less plants growing. The crop is reduced. And if this crop is reduced, that means there'll be less food for the people. State how the use of pesticides by farmer can increase food security for a country.
This will help kill the insects that are eating the.
Excellent, excellent, Tola. It will kill the insects that are eating the crop, so therefore there will be less insects eating the crop, so therefore there will be more crop available. Good, okay? And decrease?
They
Eunice?
could also be harmful. Pesticides.
Good, they can be harmful because we do not know the long-term effects of these pesticides, and it may also kill the pests and their predators, and it can affect the plant, and that pesticide substance, chemical can come into our bodies. State 2 factors other than the use of fertilizers and pesticides. I'm only thinking of fertilization, yeah. Dangerous, yeah, dangerous, which may have led to a decrease in the percentage of food secure households. In other words, what else can cause food insecurity?
Poor farming practices.
What did they, oh, sorry, Paula. What did they say?
State.
State. They didn't say explain, no? So you only have to just name them, right? Yes?
Poor farming practices.
Poor farming practices,
I was
good.
gonna say that.
You were going to say the same thing.
Bad,
You can
bad
think of another one.
environment.
Help out another one.
Bad environment.
What about the environment?
Climate change.
Climate change, excellent. So climate change can cause it, because when climate change takes place, then it can affect the growth of the crops. I'm giving you the explanation now. Remember, they only had to state, right? And they've given us two, so we'll take it from there. So climate change, if the climate change, it leads to severe droughts and severe floods. This will destroy the crops,
Yes.
and therefore there will be less food available. What is the first one you said again?
Poor farming practices.
Poor farming practice, because the farmers are poor, are not poor, but they are not using the correct methods. For example, they're not plowing correctly, then the topsoil can get away, and if the topsoil gets away, the nutrients escape with that, and the plants won't grow properly. So that's also a problem. And overgrazing. If we overgraze, we can also take away some of that. Monoculture is another poor farming method. There's already 3 incorrect farming methods. Good. I like your answers there, guys. Right? So one is also, we can also talk about unemployment. I would rather put that last, right? I'll put that last. Increase in the size of the human population, there's a greater demand. Let's say they had to ask you to explain. Notice what I'm doing. If they had to ask you to explain, increase in the size of the human population, that is the stating part. Explaining how does this affect us, you're going to say, because there's a greater demand for food, or simply more mouths to feed. Right? Farms destroyed for development. In other words, what we were talking in the car earlier,
Yes, people building houses.
good.
Yeah.
We want to build houses, shopping centers, malls, etc. So now there was a farm there, now there's houses there. So that was supplying food. Now that place is not going to supply food anymore. Decrease in subsistence farming. Again, poor farming methods here coming in
Could
there.
also be monoculture there.
Good. Okay, monoculture coming in there. And your first one there, unfavorable environmental conditions because of climate change. Excellent, guys. I like that. Okay, so we are next with our next question now. Carbon, again with environment. I don't know whether I'm going to skip this environment question now. I want to try and cover, guys, just bear with me. I want to cover as many of the topics as possible. So if we have time, we come back to the environment questions, right? Okay, so here they're saying now, a grade 12 learner performed an investigation to determine the effect on light on the growth. I'm underlining the key words for you. On the growth of a plant, of plant shoots. The learner divided the plants that were used into 3 groups. A, tip of the shoot was intact. B, the tip of the shoot was removed. And C, the tip of the shoot was covered by a cap that does not allow light to pass through. Okay, are we happy there so far?
Yes, sir.
The diagram below shows each shoot at the start of the investigation, and next to each the same shoot at the end of the investigation. The arrows indicate the direction of light in each story there. So in A, the shoot was the tip was intact. Huh? In B, the tip you can see was cut off. And in C, there was a cap put there that does not allow the sunlight to pass through. You got that?
Yes.
Okay. Name the hormone responsible for growth response shown here.
Auxins.
Auxins, excellent. This Tola is going to get an A, I think, in the paper. Yeah? You better watch it. Explain the results in A and C. Not A and C, A and C, as illustrated. So they want the result here.
It is bending towards
Right.
light.
And there, right. So you've already told us the first thing, that in A, it is bending towards the light, right? Okay, and here?
It is growing upwards.
Upwards, right. Straight, nah? We'd rather write straight upwards. So first we're telling them the result. What was the result? This one is bending towards
Light.
the light, and that one was going straight upwards. But did they say state? They said explain.
Explain.
Yeah. Okay? So what's happening here, very quickly?
Because of the auxins, it is bending towards light. So
What happened to the auxins?
They move to the
They
to
moved
the,
to
to
the
other side
other side,
where there's no light.
causing
the
cell elongation
Cell
and cell
elongation.
division on that side. Therefore, it bends towards the light. We say it is positively phototropism.
Phototropism.
But why? Because the tip was in there. And why? Because auxins are produced at the tip. This is the whole purpose of this experiment, is to show where the auxins are. Okay? At the same time, we're also showing phototropism. Here what, all right, when we went to C, what happened? You said it grows straight upwards. Why?
It could be
Because it does not receive light at all sides.
What doesn't receive light? The whole plant?
No, the tip.
The tip is not receiving light, therefore it is not the auxins will not move because the auxins are at the tip. And because they're not receiving light, they do not move to any one of the sides, and therefore it will go straight up. So it will be equally distributed. All right, let's just see how well we did. Okay? We said the same things there. Same story, yes?
yes.
Light has no influence because there was a cap on top of there, right? Good. State 2 ways in which the learner could improve the reliability of this investigation.
Could use more plants.
He could use more plants, excellent. And another word for that, what could we say generally? Instead of saying more plants, increase. What was that thing? Sample size, right? Sample size. Good. And another one? The easier one. Come, you gave me the more difficult one.
You can repeat the...
Repeat the experiment. Remember, for reliability, what do we mean by reliability? That means anybody that does this experiment must get more or less the same result. That means we can use this result to make a decent conclusion. Let's take marking, for example. If I mark Tola's paper and Looney marks the same paper, if we get the same marks more or less, one mark this way, that way won't make a big difference. That means our marking is reliable. But if I get 10 out of 100 for Tola and Looney gets 90 out of 100 for the same paper, that means it's not reliable. There's something wrong there. A third person must mark it to see where we went wrong, right? Okay, so reliability talks about that. So repeat the investigation, use more than one plant. Name the dependent variable in this investigation.
I think it's plant growth.
Plant growth is correct, yes. Okay, growth of the plant shoots is correct, because that's the part we cannot control. We can only control whether we have the tip or whether we covered the tip, whether we are leaving the tip there, etc. State 2 factors that must be kept constant in this investigation.
Must be kept in the same place of
In the same environment, okay?
Yes.
Good.
And use the same species of plant.
We must use the same. But did they say that in this thing? Did they say that they use same species? Let's see. Where are we going? We're going right up there. Right. They just said plants, isn't it? If they said that they use the same plant, then you the same plant or the same species, you can't use that in your answer. But in this case, they were general. They said plants, so we can use it in our answer. Same species. What else? Okay, we can say that same environment in which the shoots are placed, same type of shoot is used. We can also add to this, temperature must be the same.
yes.
Because if we change the temperature, it may stimulate growth. Or remember, when we're asking for this constant, what are we saying? What are we testing? We are testing whether the auxins are at the tip and they're influencing plant growth. So if we don't make all the other factors constant, we are not sure whether any one of those other factors cause the difference. So therefore, those things must remain the same. What does this do? It increases the validity of your experiment. Anything that is dealing with the method, the correct methodology, by keeping things constant, that will improve the validity. The validity means if can we now accept your conclusion as being valid in that sense then, okay? So while reliability is more focused on the results and the repeatability of those results, validity is talking about you have been doing the experiment correctly and testing for what you set out to test for. Right, so we did one on plant hormones, right? That's about 12 marks at the end of the year. We're now moving to a section on receptors, the eye, the whole of the nervous system. I think if I stand to be correct, is about 40 marks altogether. That means the brain, spinal cord, eye, ear, all that together is 40 marks. So now we're looking at looking at the
eye.
eye.
Ha, ha, ha,
Hey,
Aslam.
it keeps us going, that one. Huh? Study the diagram below that shows the front view of the human eye and answer the questions that follow. Not the conventional, you used to seeing the other diagram, the longitudinal section of the human eye. This one is showing you the frontal eye, and that should be even easier because that is what we see. When we look at each other, this is more or less what we see. We don't see it in such detail, but maybe if you're looking closely into each other's eye, you'll see some more detail, you know. But nonetheless, there's a diagram. Identify parts A and B. Come, color A.
Is pupil.
The pupil. B.
Retina.
Not retina. Help him, Eunice.
Circular muscles, or it could be also the radial muscles.
Or
One of the
one, because we can't see the muscles.
Yes.
What we call that part? The muscles make up which part of the eye?
The iris.
There you go, it's the iris, right? So rather you say, because you can't see circular and longitudinal here.
Yes.
You can see this thing and it's the iris. Good? So it's the pupil and the iris. Describe what happens to B when light entering the eye becomes less intense. What happens to B when light entering the eye is less intense?
The radial muscles contract.
Mm-hmm.
The circular muscles relax.
Right? What does this do?
This allows the lens to be
The lens or the pupil?
the
Dilate.
pupil to dilate,
Right? And therefore, more light enters the eye. Okay? So the radial muscles of the iris contract and the circular muscles relax. Forget this 3 here. That's a mistake there. The pupil dilates and more light enters the eye. In fact, this part is not part of the answer, isn't it? Because what did they ask you? Describe what happens to B. They didn't ask you about the pupil here. Okay? They only asked you up to there. So your answer should have only covered up to that part. That was good enough. All right? Okay? Why you got 4 marks? Radial muscles contract, circular muscles relax. You must at least say once it's of the iris, because radial muscles you may find somewhere else, and circular muscles you may find it somewhere else, especially circular muscles. So you must specify of the iris, right? Now the next question says, give a reason for the change described in question 4.2.
You're
And
in a dark place, so your
so
pupil needs to dilate.
And
More light must enter
more
the
light
eye.
enters the eye. That should be the answer there, right? Okay, allow more light to enter to form a clear image. But I would actually rather say this here, because that's going to explain. That's going to explain what we are talking about. All right? Are we okay there? Tola, you all right?
Yes, sir.
You sure?
Yes, sir.
State 2 functions of the part labeled C. That's the easy one. What's part C first?
It's a
the-
sclera.
It's a sclera. Good, Eunice. That's good. It's a sclera. So what does the sclera do? Tola?
It protects the eye.
One, the most important thing, it protects all. Remember, the sclera is the part right around the eye, and it protects the eye. And another function?
It allows light to enter, or it acts as it refracts light
so
to
no.
It
bend.
keeps the eye with the fluid.
All right, you can keep the fluid of the eye inside. That may be, More importantly, it maintains the shape of the eye.
Shape.
Remember, although you have a point, what you're saying about allowing light, it's not the sclera that they're pointing to. You see, if they pointed to the sclera in the front here, because remember, part of the sclera is in the front here. That is the part that allows light to pass through. This side here is white, and the white of the eye does not allow any light, mindsetters, to pass through. It's the front part of the sclera that is called the cornea
Conjunct.
that allows light to pass through. Good? And also you can add to that, it also provides a surface onto which the muscles of the eye can attach. So there's 3 functions that you can use there. Okay? As people get older, cloudy patches sometimes form in the lens of the eye. These are called cataracts. How might cataracts affect the ability of the lens to carry out its function?
You cannot see clearly.
In other words, it forms a cloudy patch over the lens and you can't see clearly there. Most important, you can't see clearly. And I've used a very big word there, impede light from going through to the lens, meaning it doesn't allow light to pass through. Okay? A person with a cataract can have his cloudy lens replaced with one made from a special material. Now, this one here, I should have put this as a challenge question actually. State 3 properties of the material to be used successfully as a lens for a person. In other words, if we're making a new lens, what must we consider to use to make this suitable to the lens. Now you need to think about, guys, you need to think about the lens and what it does and what we'll need in this other thing that we are making. What do you think it must have? First of all, think about what does the lens do?
They allow you to see.
Allows what? How does it allow to, what does it do?
Allows light to enter the
So
eye.
therefore it must be what color?
It must be transparent.
So this material must also be?
Transparent.
There you go, you got one already. I didn't, you did it all on your own, you're so clever children. Loony? They're sharp, eh?
Yeah, very.
They're not loony, they're sharp.
Man, Aslam.
Any more?
Nice. Sir.
What else does, what's that big word we use for the lens? It's involved in what process again? With an A.
Refracting light.
Accommodation.
Accommodation. What does accommodation mean?
Binocular vision, allowing
No,
you to
no, you're
look.
going too heavy.
To be able to see near and far.
Near and far, isn't it?
Yes.
But when it does this, what must happen to the lens? What must happen to the shape of the lens?
It must be able to accommodate the
must
shape that you're looking at.
Change, isn't it?
Yes.
And if it must be changed, what can we say this material must be?
It must
What's the word?
not elastic.
Yes, it can be elastic.
oh.
Good, excellent. It must be elastic or flexible, isn't it? Good, there you got some of them already. And it must be tough. It mustn't just break easily.
Yes.
Good?
We've come to the end of today's show. Thanks for joining me and for chatting to me during the show. Please visit our website learn.mindset.co.za to get the notes and to watch the videos from earlier in the year. And remember, we are here to get you through the year, so stay connected. Goodbye.